Hello, welcome to our next lesson. We are going to look at microscope. This lesson and all the other lessons in this unit are going to focus on helping the students to develop some skills that are needed in biology. So I would refer to them as some basic skills. Remember that we have looked at biology as a subject, and then we looked at the branches of biology, the importance of biology, careers that are associated with biology. We have also looked at the life processes or the characteristics that we use to describe some things as living. And that is what we also base on to refer to other things as non-living. But this lesson is going to focus on microscope, which is a very important tool or equipment that you are going to use in biology, right? But before we get into what we have for this lesson, remember to subscribe to our YouTube channel so that you always have our videos whenever they are released. Microscope. For objectives, at the end of this lesson, the student would be able to identify the types of light microscope, the types of electron microscope, and as well magnifying lenses. So it is important for the student to have knowledge about lenses which are used in microscopes, and you know what light is. And as well, it is good for you to know what electrons are, because we are going to use all these terms in our studies today. Okay, now let's start by describing what a microscope is. Yes, microscope became very important too in biology because it was realized that there are some organisms that are too small and couldn't be seen with the unaided eyes, and so there was a need for something to magnify these organisms for observation, and that is what brought about the microscope as a tool to help us to magnify these small organisms for further studies about them. And I'm quite sure that as a student you have seen microscope somewhere, maybe in the school lab or in the hospital, right? There are so many types of microscopes, but we are going to consider these two main types. The light microscope, also referred to as the optical microscope, and then the electron microscope. These two microscopes are used to magnify specimen for viewing. And remember also that I'm going to use this word specimen quite a number of times because in biology that is what we say. So whenever I mention specimen, I mean to say the organism that we are going to observe under the microscope, or the organism that we are working on. That is what we mean by specimen, right? So we are going to pick these one by one and look at them. Of course, I've said that there are so many types of microscopes, but these are the ones that we are going to focus on: light microscope and electron microscope. So now let's start by looking at what light microscope is. The difference between light microscope and electron microscope has got to do with the first words in each case. One is light microscope, meaning that it uses light in magnifying the specimen or in magnifying the object that we want to see. The other uses electrons in magnifying, or in other words, we can say that the light microscope works by making use of light, and the electron microscope works by making use of electrons. And for light microscopes too, we can talk about two types of them. We have the simple light microscope and then the compound light microscope. Why do we refer to one as simple and the other as compound? Although they are all light microscope, meaning that they all use light. So let's start by looking at the simple light microscope. Yes, this is one of the earliest microscopes that was used in the very times when microscopes became a needful tool in biology. It is a very old one. Now we call this simple light microscope because it uses only one lens for the magnification. And you're going to understand this better when we get to the compound light microscope. So this is the lens we are talking about. So in this simple light microscope, the specimen is placed on this glass slide and it is viewed through this lens. It is only one lens for the magnification. So the specimen could be magnified about 200 to 300 times its size, and that is going to be done only by this lens. So that is why we refer to it as simple. And it uses normal light, and so it's light microscope. So as distinguished from the other, which is the compound microscope, this one does not have objective lens. Now we move to compound light microscope. Yes, this is a very common one that we see in our school labs and in the hospitals. For compound light microscope, the main point is that it uses two lenses for magnification. And what are these two lenses we are talking about? It has eyepiece lens, just like the simple light microscope, where we view through. And then in addition, this one has another lens called objective lens, which is this. So in this case, we can refer to this as an objective lens, we can refer to this as an objective lens. But you may ask, why do we have two different ones? There are even three. The other one is hidden at the back. Each of them has their own magnification. So when we talk about magnification, we're talking about the extent to which the object or the specimen can be magnified. How many times? So that is the magnification. So this one has its magnification, that has its magnification. And the other at the back also has its magnification. So this is how compound microscope works. Assuming we are using this particular objective lens, this magnifies the specimen first. Then the lens in the eyepiece would further magnify what has been magnified by the objective lens. So it's going to be something like double magnification. Exactly. So this gives the compound light microscope better magnification as compared to a simple light microscope, because the simple light microscope uses only one lens, that is the eyepiece lens. But this one uses both eyepiece lens and objective lens. So that's the difference between them. Of course, they both use light. You can see here that there is a mirror. And this mirror concentrates light through the specimen so that the specimen could easily be observed. So it means for light microscope, you can use them whenever you have the daylight or in a room where you have enough light. Or in some cases too, you can supply lights from electrical source. Good. So that is for the light microscope. We have simple light microscope and compound light microscope. Now let's go to the other, which is the electron microscope. But don't forget that we are not going to deal with the compound light microscope, all the parts of it. Yes, because as I've stated down there, it is important for you to study these parts with your teacher practically in the lab. So your biology teacher will take you through that. Now, electron microscope. Electron microscope uses beams of accelerated electrons to generate its magnified images. So, as light microscope depends on light, electron microscope depends on electrons. And they can magnify objects about 1 million times to 50 million times the size of it. So you can imagine that is quite huge. When you are talking about hundreds and thousands of magnification for light microscope, in this case you are talking about millions of magnification for the electron microscope. Here also we are going to look at two types of electron microscope. We will look at transmission electron microscope and we'll look at scanning electron microscope. Let's start with the transmission electron microscope. This is the setup. It's very huge. Yeah. And note that when the specimen has been magnified, it can be captured on the screens here. Right. Now let's look at how this one works. The main difference between the transmission electron microscope and then the scanning electron microscope has got to do with how they use the electrons in the magnification. In the case of TEM, which is the transmission electron microscope, the beams of electrons are passed through the specimen. Yes, and you can see that here. This is a specimen, and the electrons from here are made to pass through the specimen, and that is what is going to lead to the magnification of the specimen. Right. So as the image is magnified and as I've shown it on the screen, it's focused onto an imaging device. just like in this case we have shown an example like the fluorescent screen. So the point to note here is that for transmission electron microscope, the magnification is done by passing electrons through the specimen. Now let's compare that to scanning electron microscope, which is SEM. This is the setup. And how does the SEM work? Now you can see clearly here that for the SEM it's about scanning. So the electrons are cast towards the specimen, and these electrons are reflected, and when they are reflected they are detected for the image formation. This is what I mean. Electrons are cast through, then the electrons hit the specimen. When the electrons hit the specimen, they are reflected. So the electrons that are reflected are now detected, and whatever pattern that shows is the actual image of the specimen. So that's the difference between the TEM and SEM. TEM passes electrons through the specimen for the image formation, but SEM casts the electrons onto the specimen and they are deflected or reflected, and that forms an image that is captured. Yes, so that is for electron microscope. But one may ask, so why do we need the electron microscope if we have the light microscope? Yes, the point is that there are so many organisms or specimens that we cannot study with the light microscope, because the light microscope is limited in its magnification. It magnifies to highest a few thousands, but this one can magnify millions of times the size of the specimen. So that makes the electron microscope very, very important. Now we cannot end by not talking about hand lens, although it is not microscope and that is not the focus, but hand lens is also important in biology. In some cases there are some specimens that are not too small. We can although see them with our eyes, but there are some details about those specimens that we would have to also observe. For example, you're observing a moss and you see some root-like structures, but you want to see exactly how they look like. The moss can be seen with your unaided eye, but for the sake of observing the small features about them, you just have to use the hand lens, also called the magnifying lens. And that will help you to see those parts that are quite small, but they are needed. And that has been stated in here. They are important in the lab for magnifying the parts of visible specimens in order to get details. Alright. Yes, so these are what we have for our lesson. And remember that we have looked at the light microscope, we have also talked about electron microscope, and then we have looked at the hand lens. For light microscope, we talked about simple light microscope that uses only one lens for magnification. Then the compound light microscope, we talked about two lenses for magnification, the eyepiece and the objective lens. And for electron microscope, we looked at the transmission electron microscope that passes electrons through the specimen, and the scanning electron microscope that scans by casting electrons onto the specimen for them to be deflected and detected. Then hand lens, which is also very important for the specimens that although we see with our unaided eyes, but we still need to observe some details about them. So as a biology student, you should learn how to use especially the light microscope and the hand lens, because those are the ones that you'll be involved in. And yes, at your very early stages in the study of biology. Okay, so let's end our lesson by looking at these questions. State one type of each of the following: light microscope, electron microscope. Right. There is another skill that is very important for us to develop, and that is on body orientation, or what we refer to as anatomical positions. So we are going to handle that in our next lesson. Okay, so see you in that lesson. Don't forget to subscribe to our YouTube channel, as I said, so that you always be notified concerning our videos.